Thank you. Didn't realize so much of my biography was out there, but it's nice to be here. Thank you for having me. What I wanted to talk about today was really some aspects of pharmacokinetics. As you heard, that's kind of my field. And the idea was that one of the most robust kind of PET imaging studies that gets done, robust in the sense that it always works and it's very useful, is the occupancy study. Now, I was hoping to use that as a sort of a taking off point to discuss some interesting aspects of pharmacokinetics that have cropped up here and there as maybe in an appendix in some of our papers or things like that, but are kind of off the beaten trail. Okay. So first of all, in case you're not working in this field or very familiar with it, it just gives you sort of a historical perspective on what we even mean by an occupancy study. And the first kind of studies that were done by these sort of landmark studies were done by Lars Farday and colleagues in the 1980s in the Karolinska Institute in Sweden. And what they did was they scanned, in this case, people with schizophrenia with carbon-11 raclopride. Raclopride is a PET tracer that binds to dopamine D2, D3 receptors. And they scan them at unmedicated, and there's a lot of D2 receptors in the striatum, this part of the brain here that you see. And then they also scan them when they were on certain antipsychotic drugs. Haloperidol, in which case you see the signal's completely obliterated. Clozapine, which is called an atypical antipsychotic, and you see that the signal is partially blocked. And higher doses of raclopride, which itself is a dopamine D2 antagonist. And they show that the actual binding of the tracer was reduced by the presence of these antipsychotic drugs. This was groundbreaking in the sense that people had known for decades that these antipsychotic drugs, the property they all had in common was that they bind to dopamine D2 receptors, and that was probably an important part of their mechanism of action, but it was never really demonstrated in vivo, and this was the first demonstration of that. Here's another study from the same group. This time they're looking both at D1 receptors and D2 receptors. D1 was sharing 23390. And then when they gave clozapine, again, you see that there's this reduction of the binding signal that's suggestive that there's competitive interaction between the antipsychotic drug and the radiotracer. And then they did finally this other study where haloperidol, which was the kind of the drug of choice of so-called typical antipsychotics that was used for many years before the newer generation came along, had a tendency to cause sort of Parkinsonian-like effects. They refer to these as extrapyramidal side effects. And Faraday's work showed that there was a window of receptor occupancy, that if you need to occupy at least 60% of the receptors to get therapeutic efficacy, to make the psychosis be reduced. But if you occupied more than about 80% of the receptors, then you started to have these side effects, these extrapyramidal side effects. So this was really the first demonstration of this therapeutic window for antipsychotics, and it was done using imaging with PET. And this made the method be very attractive to pharma industry in particular, because it's extremely expensive to design a dose for a clinical trial, and if you hit the wrong dose and you have to adjust things, that can be billions, literally billions of dollars, and expensive. So they've adapted this kind of methodology very much, in addition to people who are just doing it for pure scientific interest. And in the interim, since that time, this is just sort of a casual search I did, and there's hundreds of studies of occupancy studies, and mostly still with antipsychotics. These are other search terms. Mostly dopamine D2, believe it or not, still the one that gets looked at the most, but also other tracer systems. Serotonin reuptake inhibitors, opioid receptors, other serotonin receptors. So there's been a lot of work with occupancy studies since that time. So they provide some insight into the pathophysiology. They provide target doses for clinical trials. And that's what makes them attractive. I'd like to talk a little bit about the just the pharmacokinetic models that go into this, in case you haven't seen this, to lay the groundwork for the other things we're going to talk about afterwards. So here's the basic competition model, and if you were at the talk this morning, a lot of this is going to look a little bit redundant, but let's just go through it. You have a mass action law, a bimolecular association and a unimolecular dissociation law, where the rate of change of the bound, B for bound tracer, is proportional to the product of the reactants, the receptor concentration and the free tracer concentration. And the dissociation process is proportional to the concentration of the bound receptor ligand complex itself, or the bound receptor ligand complex, K off times the bound. So B is the bound, R is the receptor concentration, F is the free ligand concentration, K on is the association rate constant, K off is the dissociation rate constant. You can write this down as a differential equation, and you get the basic competition model. It's a competition between association and dissociation. At equilibrium, if you solve for the bound, you find that the bound equals Bmax, that's the total amount of receptors that are available, times the free concentration over the free plus this ratio of the off rate to the on rate. The off rate to the on rate is commonly called the equilibrium dissociation constant, the KD. So if we replace that, you get the bound equals Bmax times the free over the free plus the KD. And you can separate these into a product of the Bmax, the receptor density that's available, and this other term that's dependent only on the tracer concentration, the free over the free plus the KD. This is called the occupancy, because you can see in this form that it shows you the fraction of the receptors that are occupied by the ligand. Bound, Bmax times the occupancy. Okay. So here's the occupancy, and it's useful to rewrite this a slightly different way, which is to write the free in relation to the KD. Write the free in unitless or in KD units, and then you can see that the fraction of receptors that are occupied is purely a function of the free concentration relative to its KD. There are no other factors. It's only the amount of free ligand relative to its own KD for that receptor site. Absolute concentration is not relevant at all. Okay, what about if there are multiple competing species? That's really the case we're going to be interested in. They all follow their own mass action laws. Species B1 through Bn, and each one of them is following the identical mass action law as the single species case, except now the receptors that are available is Bmax minus the sum of all the bound, okay, because there are all these different competing species that are bound. And if you run through this and you do equilibrium and you run through the algebra, what you get is that the bound of the jth species is Bmax times the free of the jth relative to its KD over 1 plus the sum of all the frees relative to their KDs. So that's the occupancy in the case that you have multiple competing species. We're going to be interested in a very special case, which is that in PET imaging, the radio tracer itself is always a very low concentration, which we call tracer dose. And that means in particular that the radio tracer concentration is much less than its KD. And we're only interested in one competing species, an exogenous drug that we're given, and we want to measure its occupancy. So in that case, we have free, F for the free tracer, D for the free drug, and the drug occupancy is going to be the drug occupancy relative to its KD over this sum of 1 plus the free tracer relative to its KD plus the drug relative to its KD. But because of the tracer dose condition, this is a negligible term. It drops out, and what you were left with is that the drug occupancy is the same as it would be in the absence of the radio tracer. And that's the whole philosophy of doing tracer dose experiments, is that they don't perturb the system. Its concentration is so low that all you really see is the same occupancy the drug would have in the absence of the tracer. Okay. How do we extract that from a PET study? Well, usually we do a series of scans. We do a baseline scan and we extract a parameter called the binding potential. And again, I don't have time to really go into how we derive it. We talked about it somewhat this morning, but suffice to say that it's proportional to the receptor density, Bmax, divided by the KD of the tracer. And when you do the same experiment with a drug on board, the effective KD is reduced, and the factor it's reduced by is 1 plus the drug concentration relative to its KD. And then if you finally take the relative change of the binding potential between the baseline of the drug condition normalized to the baseline condition, what you wind up with, algebraically, is just the actual occupancy by the drug. So you can extract the occupancy of the drug by doing a baseline scan, doing a second scan while the drug is on board, and then taking the relative change in the binding potential. And that's the parameter that we're interested in drug occupancy studies. Okay, if you do that at a number of different concentrations of the drug, you can get a number of different concentration points of the drug occupancy. And if you plot the log of the competitor concentration, some metric of the competitor concentration, either the actual concentration in the plasma, if you can measure that, or even just the dose, if you know what the dose you're administering is, against the occupancy, you get this sigmoid curve, an occupancy curve, and you get some measure or a metric of location called the EC50 or the ED50. EC50 for the expected concentration that will give you 50% occupancy, or ED for the expected dose that will give you 50% occupancy. So that's the measure of location of the occupancy, of the propensity of the drug to occupy the receptor. Okay, so that's the basic setup. That's what we're working with, the theory. And here is an example I'd like to show you. And that's a study in which we were measuring the occupancy of dopamine D2, D3 receptors by a drug called aripiprazole. Now, aripiprazole is a new generation antipsychotic drug, and it acts as something called a partial agonist, which is to say that rather than just blocking the action of dopamine like most of the antipsychotics do, they're called antagonists, it actually mimics the stimulating effect of dopamine, but it does so weakly. So it displaces the dopamine, it allows transmission to continue, but it does so at a lower level. And the result is that there is a claim for aripiprazole that you can occupy a much higher fraction of the receptors than you can with the typical and atypical antipsychotics without getting extrapyramidal side effects. So we were looking at this drug at therapeutic doses. We're using a tracer called F18 labeled falipride. It's a dopamine D2, D3 tracer, and it has the property that it gives you a good strong signal in the striatum. This is the somatomotor-sensory striatum, the associative striatum, the ventral striatum, which is really the nucleus accumbens. And it also gives you good signal in some extra striatal regions. Down here you can see the thalamus and the hippocampus. It's a little controversial if you can get a good enough signal in the cortex, the dorsolateral prefrontal cortex, but certainly in these limbic regions you can get a good signal. And then here's the cerebellum. It's our reference tissue. It's a tissue in the brain that has negligible amounts of D2 receptors. This is the tracer we were using. These are binding potential maps, actually, to give you an idea of what it looks like. This is in the in the striatum, in a coronal view, and then in the striatum in a transverse view. But then here you can also see in these extrastriatal regions. This is the amygdala over here in the transverse view. This is the temporal cortex, the temporal lobe. And then over here you can see the thalamus. This is the tail of the caudate and the tail of the putamen, but right in the center is the thalamus. So you can get these good strong signals in these extrastriatal regions as well. You pay a price for that. You have to scan for 4 hours to be able to get the striatum and the extrastriatal regions, but you can get them all simultaneously. Okay, so here's aripiprazole. Here's the study we did. We scanned 17 patients with schizophrenia who were all on therapeutic doses, but they were on a wide range of doses. The doses ranged from 2 milligrams to 40 milligrams. So we really had a broad range of doses to be able to map out the occupancy. Okay? And here's some sample data. This is the average binding potential in 9 subjects at baseline. And you can see as the doses get higher and higher that the binding potential gets lower and lower. It's doing what we expected to do. There's a dose occupancy relationship. It's all in the striatum. Here at the highest dose is 30 milligram, and it's virtually 100% blockade. There's no binding potential left at all. And when we made those sigmoid plots, we saw the following thing. We saw in blue the striatum, and in the orange here the extrastriatal regions. That is, the measure of location was smaller for the extrastriatal regions. The EC50 was smaller, the ED50 was smaller, or another way of saying it is that the aripiprazole appeared to bind more strongly in the cortex or in the limbic regions than it did in the striatum. Okay. Question we wanted to answer was, is this a real pharmacological effect? Is there really different occupancy in different parts of the brains? Or is this possibly a kinetic artifact? And to understand this, let me remind you about the models we're using. It's always this two-tissue compartment model in which you have the concentration in the plasma, CP here. It goes into the brain. We call it CND for non-displaceable because it's the sum of the free tracer and the non-specifically bound tracer. And then the pink compartment here is CS, that's the specifically bound. That's what we're interested in. That's where the binding potential resides. And going with these is a set of coupled ordinary linear differential equations. And we can extract the binding potential from as a function of these rate constants. It's K3 over K4, actually. And the assumption is that all of these are constant coefficients. During the time of the experiment, these coefficients are all assumed to be non-changing. In particular, K3 is proportional to Bmax, the receptor density. But what if that's not true? What if you have a case where the occupancy is actually changing over the course of the experiment so that the apparent K3 is no longer constant? It's the original K3 times 1 minus the time-changing occupancy. So here's the setting. We're looking at a drug occupancy study at a target receptor. And we're doing a bolus PET experiment. And we're looking at the two-tissue compartment model. It's observed that the percent decrease in binding in these different regions is different. And he's putting it sort of didactically here. Can it be inferred that the drug has different occupancy at these different places? The answer is not necessarily, especially if the regions have very different Bmax. Now, you recall from the earlier slide that that is the case for dopamine D2 receptors. They're very high density in the striatum. They're order of magnitude lower density in these extrastriatal regions, like the hippocampus and the thalamus. Okay, so this is a hard thing to study experimentally, other than what we observed, and we don't really know why what we observed happens. So it's a good situation to at least do a simulation to see if you can understand this at a theoretical level. The setup is this, that the tracer is at tracer dose. At each time, the drug occupancy is approximately what it would be in the absence of the tracer, because the tracer is a tracer dose. Receptor availability is 1 minus the occupancy times what it would be otherwise. And this leads to a set of ODEs, still linear differential equations, but now they have time-varying coefficients. And this has an effect on our outcome measure, because our outcome measure is based on fitting the data, assuming that the coefficients are constant. Okay. Here's the system. Here's the specific binding equation. Now, because it looks like this, we're only looking at this part, right, between here and here. And the rate of change of the specifically bound is just this function of the non-displaceable concentration and the specifically bound itself. And formally, you can write that down as a convolution integral. That's the solution to this convolution integral. Now, in reality, we know that the non-specifically bound is itself affected by this. So it's not as if this is an unperturbed input. This is not necessarily an unperturbed input to that system. But formally, we can write it down that way. And it's useful because it's going to lead us to understanding what the relationship is here. Okay? There are several ways of writing down the binding potential. And one of them is, as we discussed earlier this morning, a ratio of areas under the curves, or ratios of integrals to infinity. You don't ever really measure this because your PET experiment doesn't go on to infinity, but theoretically it's convenient to write it down this way, and it is numerically equivalent to any other way you might write down the binding potential. Okay, so I'm going to just take you through this. I know it's a little dense in the equations for a slide presentation, but it's fun and interesting enough, I think, that it's worth looking at these steps. Here is the relative change in the binding potential. 1 minus the binding potential in the drug condition divided by the binding potential in the baseline condition. If you write that down in the integral form, You have 1 minus this ratio of integrals. Again, it's the specific binding compartment divided by the plasma concentration, drug condition on top, baseline condition on the bottom. I hope you can see down here, if we can assume, and this is a thought experiment, so we can assume whatever we want. In this case, what we'd like to assume is that the radio tracer injection was identical across the two scans, so that we can eliminate it from this equation. Now we've reduced it to 1 minus the ratio of the specific binding integral in the drug condition divided by the specific binding integral in the baseline condition. Okay. What is that integral? Well, here it is formally. Remember we said it's this convolution. And that means that we can write it down as a product of two integrals. The part that's associated with the non-specific compartment, the input, and this exponential, e to the minus k4t, and we can integrate that out. We know what the answer to that is, just 1 over k4. So at the end, we get that the specific binding integral is k3 over k4 times 1 minus the occupancy times cnd over time. The point being, at this point, we've eliminated any reference to the specific binding itself. It's all a function of the non-specific binding and the occupancy. Okay? At baseline, it looks like this because there's no occupancy. In the drug condition, it looks like this. 1 minus that ratio is this. And now there's another fact about the kinetics that we can use, and that is the integral of the non-displaceable compartment is the same under any conditions. And you can show this mathematically, and you can show it experimentally. It's not perturbed by the fact that there's occupancy on board. It's the same under both conditions. And that allows us to simplify this even one step further. At the bottom, we have that the delta BP, the fractional change in the occupancy, is really this weighted sum of the occupancy over the course of the experiment, the weight being given by the relative concentration in the non-displaceable compartment during that time. Okay, here it is. Okay, so also you can see, by the way, as a reality check, if the occupancy had been constant during the experiment, it would just come out of here. You'd have, the numerator and denominator be the same, and it would just equal the occupancy. So it's in the boundary case, it makes sense, right? But for this to have any impact at all, you have to have a case where the drug occupancy is changing over time. We don't even know if that is the case. Well, with aripiprazole, actually, let me step back and say something different. It's changing over time, but then the question is, well, why should that be different in different brain regions, right? Because it's changing over time, but wouldn't that give you the same answer regardless? And the answer is no, and the reason is this. Imagine you have this, here's your striatum with a lot of receptors. Here's your hippocampus with a small amount of receptors. This is a capacitance for soaking up a tracer when you do your experiment. This has less capacitance for soaking up tracer. And what it does in effect is it buffers the non-specific or the non-displaceable component. So you can imagine sort of in stop time, first you get tracer in the plasma, it gets into the brain, it's about the same concentration in both of these regions, but then it starts to bind, and this is a bigger sink than this is, so that this free concentration is actually higher than this free or this non-displaceable concentration because more of it has bound to the receptor. That continues to happen. Then when the kinetics reverse and they're dominated by efflux rather than influx, what happens is now you have this whole sequestered pool that was a sink that becomes a source. And suddenly this gets lower in the late part of the experiment, but this stays elevated. So they cross, the two things cross. And that shows a way that you can actually have different concentration over time of the non-displaceable compartment. It's important to realize that the integral will be the same. In the two different compartments, the integral of the non-displaceable compartment over time is identical. But its actual kinetics, its actual concentration over time is different, with it being higher in the low binding regions early and then higher in the high binding regions later. Okay, here are some time activity curves using phallipride, and these are simulated curves, but the simulated curves using a real input function, real plasma concentration, and the extracted parameters, the extracted rate constants from real phallipride experiments. This is the striatum. Here's that non-displaceable compartment down here. The dotted line, the dashed line rather, is the specific binding. The solid line is the sum of the two. That's the observable. That's what you can actually see. Here is the hippocampus. Same thing. The dotted line is the non-displaceable compartment. The dashed line is the specific binding compartment. The solid line is the sum of the two. Down here, I hope you can all see down in the corner, I've just put the two non-displaceable curves. The orange one is the striatum. The green one is the hippocampus. And you can see that early on, hippocampus is actually higher concentration. At some point, it's maybe a little hard to see if they cross, because they have to cross because they have the same integral. So the high binding region has to make up for it later on. Here's the same two things with occupancy, hypothetical occupancy, just to show that even though the receptor availability becomes less due to the occupancy, you still have that phenomenon, that it's lower in the high binding region early on and higher later on. Okay. Is this something that really is plausible to be happening with aripiprazole? This is from a paper from Malikarjun et al., where they were just looking at the PK, the plasma concentration of aripiprazole in healthy controls over a 24-hour period. And they wanted to measure these pharmacokinetic parameters of aripiprazole, in particular things like the half-life, the maximum concentration, and so forth. And what you can see is that even though the total half-life is quite slow, something on the order of 40 hours, that it probably is not constant. It's probably multi-exponential. And if you just sort of eyeball this part in here, that's the first 24 hours after the dose administration, the washout rate really is faster, and it corresponds to a half-life of something on the order of 15 to 20 hours rather than 40 hours. So it is conceivable that there is actually washout happening during the course of this long 4-hour Falley-Bright scan. So we did some simulations. Simulated two regions, the striatum, where the binding potential is 22, the hippocampus, where the binding potential is 1.4, generated a 4-hour hypothetical scan, had exponentially decreasing drug occupancy with 3 different half-lives, 600, 900, or 1,200 minutes, and there was in the 10 to 20 hour range. And then for each region and initial occupancy combination, we tried different initial occupancies, 60, 75, and 90%. Did 1,000 simulations with added noise, Monte Carlo simulations, and then let the fitting routine fit the data and see what it told us the occupancy was. Here's the results. So here's hippocampus always in blue bars, striatum always in orange. The unweighted average of the occupancy over the course of the experiment is in, well, it should be green. It's kind of mustardy looking here. And the initial occupancy, the starting value, is the dotted line. And just to give you some feel for the change that happens in the occupancy, with a 600-minute half-life, it decreases by about 24% over the course of the experiment. 900 minutes, 17%, 1,200 minutes, 13%. And what we see is that at each combination, the hippocampus has a higher occupancy, a higher apparent occupancy, that corresponds more closely to the initial occupancy. Here's hippocampus at the 3 different half-lives. Here's the highest, 90% initial, 3 different half-lives. Whereas the striatum is much more closely aligned with the unweighted average. That's close to the average occupancy over the course of the experiment. So the simulations at least show that, yes, this is a plausible kinetic artifact that could be happening. So the summary for example 1 is that a kinetic artifact is a plausible explanation of the apparent regional differences in occupancies. It's not necessarily the case that occupancy is different in different brain regions, and when you see papers that claim it's different in different brain regions and then have a very long discussion section about the psychiatric implications of that, keep in mind that PET measurements have their limitations. Okay, example 2. Suppose we have a case where the drug and the tracer both have a non-negligible affinity for more than one target molecule. Can we measure occupancy? Now, everything I've shown you up till now, there's always one target receptor. Both the tracer and the drug are going for that one target receptor. And there's a very specific case we had in mind because we had to deal with this. And that has to do with measuring dopamine D3 receptors. Now, the D3 receptor is in the D2-like family. It was discovered by Pierre Sokolov in 1990, I think. He first characterized it. It's very similar by protein, peptide homology. They're very similar structures. But it's located in different parts of the brain, and it's pharmacologically quite distinct from the D2 receptor. In 2005, Alan Wilson is a radiochemist in Toronto, synthesized a tracer called carbon-11 PHNO. He developed PHNO originally because it was an agonist. So there was some interest in the field at the time of having agonist radio tracers rather than antagonists. And that was his purpose for developing PHNO. But in the literature, there is some controversy as to whether PHNO is more selective for D2 or D3. In some papers, it was suggested that it was strongly selective for D3 compared to D2. And the first papers that came out of the Toronto group, that was in fact the case. So this is a human brain. Here you can see that the strongest signal is in the globus pallidus. And if you look at this in terms of time activity curves, you see that the pallidus signal takes a lot longer to wash out than in the dorsal stratum, like the putamen back here. This is a paper from Murray et al., an autoradiography paper. It may be a little hard to see this, but the point is this. They did autoradiography of D3 receptors, and the D3 is most highly expressed in the nucleus accumbens, that's the ventral stratum, and in the pallidum, and also to some extent in the substantia nigra, in the midbrain. But the fact that we saw this, that people were seeing this high signal in the pallidum was completely consistent with PHNO being a D3 preferring radio tracer. But it also binds to D2. Okay? So just to give you a sense, here is the D3 receptor distribution. This is in the nucleus accumbens and the pallidum down here, and down in the substantia nigra. It's very, very small, particular parts of the brain where it's distributed, and it's low density. It's never as much as D2, any of these places. This is where it's the highest, but that's still not anywhere near as high a density as the D2 receptors. Okay. So our lab did a series of experiments with PHNO, first to verify and validate the fact that this was really a D3 signal we were seeing and that the tracer is D3 preferring. We scanned non-human primates, baboons, with PHNO at baseline, and then we gave a competitor. BP897 is a dopamine D3 preferring partial agonist. So it's about 80-fold, 70 to 80-fold selective for D3 over D2 in vitro preparations. And what you can see is that when we gave BP897 at this particular concentration, 0.25 milligrams per kilogram, that the pallidum signal here, this high signal in the pallidum where there's a lot of D3, gets completely blocked. It's gone. But the dorsal stratum here is largely untouched. The D2 part of the signal is largely untouched. And this is validation that that PHNO really is D3 preferring. Here, for comparison, is a raclopride scan in the same animal. Raclopride is not D3 preferring, so you can see at baseline it doesn't even have this strong D3 component. So after you give BP897, the PHNO looks like raclopride, essentially. So we have a tracer that's got some affinity for these two different receptor sites. Here's time activity curves in the monkeys. This is predominantly the D2 part here. This is the D3 dominated part in the pallidum, the thalamus, and the substantia nigra. Here's after BP897. You can see that the D3 rich regions collapse kind of onto the D2 regions. Okay. Here's the binding potentials. The red is raclopride. We're not that concerned with that. But the point is it's very high in the pallidum, moderate in the ventral stratum, lower in the dorsal stratum, and somewhat lower, but still quantifiable in the substantia nigra and the thalamus. When we give the BP897, you see the highest occupancy, the biggest change in the pallidum. We can't really legitimately call this occupancy at this point because we know it's a mixed signal. We don't know what's binding to what. But also, we know that in the substantia nigra, you block almost the whole signal. We block all of the signals, so that all of the binding of PHNO in the substantia nigra is due to the D3 receptors. So with our colleagues at GlaxoSmithKline, who were also interested in this question, we undertook another series of experiments. There was, first of all, one small criticism of that first set with BPA97 is that it's a partial agonist and it's not extremely selective for D3, but GSK had a more selective compound, SB277011, which is more highly selective for D3 and an antagonist. So it's really a better, cleaner compound to do this kind of experiment with. So the design was to give PHNO, do a baseline scan, start an infusion of the SB277011, and continue that infusion through a second PHNO scan. We were able to do that at a number of different doses of SB277011, 0.5 milligrams per kilogram, 2 and 5 milligrams per kilogram. And also we had another interesting tool compound to work with called SV156, which is the opposite. It's selective for D2, not D3. And here are the results. Again, these are binding potential maps, and you see very similar images to what we saw with the BPA97. High binding in the pallidum at baseline, which gets completely blocked in the presence of high concentrations of SB277011, and then just the opposite, the mirror image, what you would expect with the SV156. This is the D2 selective compound. It spares the D3 part of the signal but blocks the dorsal striatum where the D2 signal is. Okay, so we wanted to try to make some kind of quantitative model out of this to see if we could disentangle what's the D3 part of the signal, what's the D2 part of the signal. And we thought of it this way, that the total binding potential you observed in each brain region was decomposed into the D3 fraction, which we're symbolizing here with FD3 for the D3 fraction, and the D2 part, which is 1 minus FD3, and that when you saw a change across conditions, the relative change in the binding potential, that you could also write this as the same weighted sum, the D3 fraction times the occupancy at the D3 receptors, plus the D2 fraction times the occupancy at the D2 receptors. When we did that and we did a regression analysis to estimate these D3 and D2 fractions across these different regions, we saw that the D3 fraction, the estimated D3 fraction, was nearly 100% in the midbrain, the substantia nigra VTA, about 70% in the pallidum, 80% in the thalamus, 50% in the ventral striatum, and negligible in the dorsal striatum. So very consistent with the autoradiography literature about where the D3 is. But keep in mind, this is not saying what the D3 density is in those regions. This is saying what fraction of the PHNO binding potential is associated with the D3 receptor. Okay. Since that time, we've done that a lot more. We've done it in different contexts in rhesus monkeys. The GSK group did it in humans, and what we find is that this D3 fraction is very conserved across primate species. So these first two are two different models, but they're both in rhesus monkeys. The second two bars are the two different models in the baboon, and then the blue is the human. And you can see there's nearly 100% of the PHNO binding potential in the substantia nigra is due to D3, a moderate, more than 50%, but somewhere less than 80% in the pallidum is D3, 50% in the ventral striatum, again 70 to 80% in the thalamus, and negligible amounts in the dorsal striatum. Okay, so that's where it stood. That was interesting, but the question becomes, why is this important? How are we going to use this? And it became relevant when this study was published. Now, the Toronto group where Alan Wilson works are the ones who've used PHNO the most, not surprisingly, because that's where the compound was developed. And they scanned patients with schizophrenia on different antipsychotic drugs and compared them to control subjects that were drug-free. And what they saw, here's the putamen on the left and the pallidum on the right. Here's the controls, leftmost, and these are binding potentials, a scatter plot across the subjects. These are patients on olanzapine, clozapine, and risperidone, 3 different atypical antipsychotic drugs. And in the putamen, the dorsal striatum, where the signal is predominantly D2, they got the expected effect, which is that you got a decrease in the binding potential that you could associate with a occupancy of those receptors by the antipsychotics. In the pallidum, they got a paradoxical effect. Here's the controls. Patients, again, on olanzapine, clozapine, and risperidone, and they actually got an increase in binding across the conditions. Now you can see there's some outliers here, but even when you get rid of the outliers, if you kind of look at the medians of these, you still get this effect that at the most you can say that there's no decrease in the binding potential. And that raised a number of questions in our mind, which is, A, is it really the case that these drugs don't bind to D3 in vivo? That's one possibility. Another possibility is, again, it's a kinetic artifact because it's difficult to measure PHNO in the pallidum. It's not as easy as it is in the striatum, so consider that possibility. And then the third possibility is that in response to the antipsychotic drugs, that D3 receptors are upregulated. So this is actually an artifact of the fact that after taking the drug for a while, the patients had higher D3 expression than the controls did. And we wanted to see which of these were the case using PHNO imaging in non-human primates. This is the study we did, and this is a table from that study, which is taking these drugs, clozapine, haloperidol, risperidone, olanzapine. They're all the antipsychotics that get used. This is their D2 to D3 selectivity in vitro, in vitro assays. And in every case, they're more selective for D2 than they are for D3. So it shouldn't be surprising that they bind more to D2 than D3. But if you look at the typical occupancies, the target occupancies that you want to get with these drugs, it's a little lower for clozapine, 60%, 75% receptor occupancy with these other drugs. And then you look at the predicted occupancy at D3 receptors at the same level of drug that will give you this occupancy at D2, and given these selectivity ratios, it should be substantial. Less than at D2, but still it should be substantial. So there was something paradoxical about that Toronto study. And to analyze this, we scanned 3 baboons. We scanned them at baseline, the following antipsychotic drugs. We used haloperidol at 0.01 milligrams per kilogram. We used clozapine at 0.5 milligrams per kilogram. In both cases, the drug concentration was selected to try to get the D2 occupancy that is the target occupancy in humans, 75% for haloperidol, 50% for clozapine. Each subject was studied twice. And here are the binding potential maps as a result. First, you can see at the level of the pallidum, again, that there certainly is some reduction. It may not be projecting that well, but there's certainly some reduction in the pallidum, both with clozapine and more obviously so with haloperidol. These are following acute doses, by the way. The patients would have been on chronic dosing, so they would have had time for this upregulation to occur. In the monkeys, they're just getting a single injection of the compound, so there's no time for this upregulation to occur. So we're just looking at the acute effects of binding of the drug. And here are the delta BPs in all the regions. Blue is clozapine, orange is the haloperidol. We applied this same model that we used before, but using the fact that there's this strong species homology and that it really seems to be pretty consistent across subjects, what we did was to fix the D3 fractions in this case and just measure the occupancy of the drug at D3 and at D2. We did a regression analysis on this model to extract what the occupancy of the clozapine and the haloperidol were at these different, all these different brain regions, and then use that to measure the in vivo selectivity. Okay. Here's the results. At D2, we got 47% occupancy of clozapine. At D3, 15%. So again, less than at D2, but not negligible. Some definite occupancy. With haloperidol, it was a lot clearer. We got 76% on average in the dorsal for D2 rather, and 57% at D3. So the in vitro selectivity ratios are about 3 for both of them. In vivo, we got something that's not quite identical to that, but enough to suggest that acutely these drugs do bind to D3 receptors, so that what was observed in the patients was some other phenomenon, either a kinetic artifact or upregulation of the receptors. So in this case, the summary of this example was that the antipsychotic drugs do bind acutely to D3 receptors, and they do so with a D2, D3 selectivity ratio that's comparable to the in vitro setting. We needed to do an occupancy study, but with multiple targets. Okay. I don't know about time here. I do have one other example. It's a little shorter than the others. Maybe we can run through this. And that is that everything we looked at so far are using an exogenous drug to look at its occupancy. But this competitive binding technique has been used for a long time with challenges, pharmacological challenges, that induce release of the endogenous neurotransmitter, in particular using amphetamine to release dopamine. And it's the same technique. But the question is, are we looking at occupancy, or are we looking at some other phenomenon besides occupancy? Here's the basic setup. At baseline, you have the tracer. It binds to the receptors. You give amphetamine, and amphetamine induces release of dopamine at the synapse. The dopamine competes with the radiotracer, and you should get a reduction in the binding potential, again, by exactly that same competitive model that we were just talking about in all the cases with the occupancy. Unfortunately, the data didn't support it very well. There was a number of studies that showed that the reduction in the binding potential stayed suppressed, it stayed lower for a long period, much longer than the surge of dopamine. And this was shown with microdialysis studies, where the dopamine surge was shown to peak at about 20 minutes to 40 minutes after the amphetamine injection, and pretty much subside to baseline by about 2 hours. But here's a study from our group where we looked at baboons at 1 hour after the amphetamine injection, 4 hours, and then 24 hours. 24 hours later, and 48 hours later. So the point is the binding potential stays suppressed for a lot longer than can be supported by the idea that the tracer is just competing with the dopamine. And to try to explain this, Mark Larawal suggested the internalization model, the idea being that when you stimulate the receptors, that a number of them internalize, and that perhaps the radiotracer is unable to bind to those internalized receptors. To test this, we collaborated with the labs of Jonathan Javich and Steve Raport at Columbia, and they came up with a really very elegant set of experiments. It's not our experiments, it's theirs, but really quite elegant. And the idea was this: we measured the binding at baseline. You give this is in a cell preparation, by the way. They did this in transfected cells, kidney cells transfected, human kidney cells transfected with D2 receptors. You give a dopamine agonist, that's quinpirole. It induces receptor internalization. You give a compound called MT-set. Now, MT-set has the property that it binds covalently to these surface-bound receptors. It doesn't internalize, but it binds covalently at the surface. It both fixes these receptors to the surface and inactivates them. They're unable to bind dopamine on the surface anymore. And then you measure the binding again, and under this condition, you can be pretty sure that what you're measuring is binding to internalized receptors. And when they did that, what they found is that almost all of the major dopamine D2, D3 tracers did internalize, and they bound to the internalized receptors, but they did so at a much lower affinity. The KD was, on average, doubled for the internalized receptors compared to the external receptors. For example, 0.5 in the externalized case for raclopride, 1 on average in the internalized case. So this would be quite consistent with the reduced binding staying down for longer if this was to internalize receptors. We still had no direct evidence, however, that these receptors were really internalizing, that this is what we were seeing. And to answer this, a doctoral student in Dave Sibley and Bob Innes's lab at NIH did another series of very elegant experiments. This is Mette Skinchberg. Since then, she's joined our lab at Columbia. And what she did was look at radiotracer binding with amphetamine in wild-type and arrestin knockout mice. These are arrestin-2 knockout mice. Arrestin is a protein that's essential for internalization. So these mice are unable to internalize their D2 receptors in response to dopamine stimulation. It's just a cartoon showing that they can't internalize because they don't have the arrestin-2. And what she observed, this is with two different tracers, carbon-11 MNPA, which is a dopamine D2 agonist, and falipride as before, was here's baseline binding potentials, here's 30 minutes after injection, and both the wild type and the knockout mice stayed suppressed, have reduced binding at 30 minutes like you'd expect, but 4 hours after the injection, the wild types were still mostly suppressed. They had mostly reduced binding potentials, whereas the knockout mouse came back to baseline. This was with both tracers. So this was very strong and very elegant evidence that probably what this phenomenon is due to receptor internalization. So the summary of example 3, the amphetamine effect is likely to be predominantly competition with DA in the early phase, but predominantly internalization in the late phase. This results from two different ways of altering the affinity. One is the apparent occupancy by the dopamine. The second is a real reduction of affinity due to the internalization. Or, in other words, occupancy does not always account for everything. Okay. And in closing, I just want to acknowledge all the collaborators I've had the pleasure of working with over this time. In our division, translational imaging, and this Abhi Dargram is the director. Lots of alumni have passed through over the years. Here's our fellows, here's our GSK collaborators, and our technical staff who helped us quite a bit with all these experiments. And I thank you for your attention.